Hi everyone, my name is Linda. I am a senior at Georgia State University, majoring in chemistry with a concentration in biochemistry, and I am presenting a study of near-infrared DNA damage and photo-cytotoxicity by a brominated 4-quinolinum dicarbocyanine dye named ESS24. My research focuses on photodynamic therapy, or PDT, which is a form of therapy in which a photosensitizing agent generates reactive oxygen species in cancerous tissues. This process will damage DNA and other macromolecules, which will lead to cell death. PDT is effective against ailments for which surgery is not an option. Many PDT agents usually absorb red light. This is because this wavelength window allows the light to reach deeply into cancerous tissues. Here, I work on a specific dicarbocyanine dye called ESS24. ESS24 has demonstrated through its characteristic that it could be a potential candidate for PDT. It is also taken up by ES2 ovarian cancer cells and causes a substantial level of near-infrared photo-cytotoxicity. ESS24 is composed of 2 aromatic rings which contain nitrogen bonded to methyl groups. The 5 carbon chains that links the 2 aromatic rings containing the nitrogen has a bromine at the center, and so it is mesobrominated, and it's also conjugated, as you can see in figure 1. The point of my research is to establish the fact that when irradiated with infrared light, the dye will show enhanced photodynamic activity in the presence of DNA as well as in ovarian cancer cells. Methodology for this research included determination of the specific wavelength or the range of wavelengths under which ESS24 will display optimal photonuclease activity. The principal methods used were UV-vis absorption spectroscopy, DNA photocleavage, and photocytotoxicity experiments. The expected results were to find enhanced activity of the compound upon its irradiation, the type and mechanism of the ROS being generated upon photocleavage by the dye, and the precise DNA binding mode of the dye. The UV-vis spectroscopy experiment showed that the ESS24 is stable in sodium phosphate buffer in the absence or presence of DNA, as you can see on figure 3 and 4. For this result, we can conclude that ESS24 was able to display minimal absorption at long wavelength value. However, when the dye is by itself and mixed with the DMSO solvent, it displays a much stronger absorption, as you can see on figure 2. For the DNA photocleavage experiment, cleavage can be observed in lane 2 here, or figure 5, which is the lane that contained a sample made of the dye, the sodium buffer, DNA, DI water, and was irradiated under the light at 830 nanometer at 10 degrees Celsius. However, when the sample was kept in the dark, no cleavage of DNA was observed, as you can see on lane 3 and 4. These results demonstrate that the ESS24 is capable of cleaving double-stranded DNA under the given condition, which shows potential as a photosensitizing agent in PDT. In the photocytotoxicity study, the conclusion reached were that the ES2 ovarian cancer cells can take up the ESS24, as you can see in figure 7. The percentage of survival of those cells was greatly reduced, as you can see in figure 8. And last but not least, there was a significant amount of ROS production when the dye was irradiated under long wavelength light, as you can see on figure 9, compared to when there was no light, as you can see on figure 6. So in conclusion, our findings demonstrate that the ESS24 dye may be a potential candidate as a photosensitizing agent for PDT, as it would produce hydroxyl radical. I would like to give special acknowledgement to my faculty sponsor, Dr. Grant, our faculty collaborators, Dr. Henry and Dr. Taratula, as well as my mentor, Effie. I would like to thank the Honors College for allowing me to pursue research at Georgia State University, from which I discovered this conference. Thank you, and please feel free to give me any feedback during the online poster session on Saturday.